Today we are going to learn about the sexual reproduction in plants. Flower is the reproductive organ of a plant. Okay. So here we are able to find a bisexual flower is given to you. In that there are 4 whorls. The total floral parts will be arranged in 4 different whorls. See, on the pedicel there is a platform called thalamus. Just above that we are able to find there is a whorl called calyx. We are calling them as sepals. Next one is the green color one, it is called as sepal or the calyx, and the red color one is called petals or corolla. Next here we are able to find the stamen. Stamen means yellow color bob like structure it is there, and filament and anther both the parts will be there in the stamen. And you are able to find the gynoecium, which is having 3 parts. The basal part is swollen, which is resembling like a flask like structure called as ovary, style and stigma. 3 parts will be there. So these are all 4 whorls are there. We can say that one has a complete flower, and it is having gynoecium and androecium both the parts, so it is called a bisexual flower. Next here we are able to find the first diagram reveals that it is a bisexual flower, which is having androecium and gynoecium both the parts are there in the flower, so it is a bisexual flower. Mostly the self pollination is opted here. Next, second diagram indicates it is the flower is having only the stamen, but it is not having the gynoecium part, so it is a male flower. So next third diagram, if you observe, it is the female flower which is having only gynoecium, and stamen will not be there here. Male parts are not there. So here there is an option for cross pollination. Pollination means the transfer of pollen grains from anther to the stigma is called pollination. Now here the male gametes are pollen grains. If you observe the pollen grains by dropping the flower pollen grains in a small china dish with water, if you drop them and if you observe under the 1 drop of water with pollen grain under the microscope, you are able to find the outer coat is little bit spiny in nature, inner coat it is smooth, and it is having 2 nuclei. One nucleus is called as vegetative nucleus, which is responsible for the formation of pollen tube, and the other one is generative nucleus, which divides and then 2 male nuclei will be formed. Now in the case of flowering plant there is a double fertilization. Fertilization is nothing but the male gamete fuses with the female gamete. Here the male gametes are the pollen grains which are dropped on the stigma of the flower. So they start germinating. All the pollen grains they may not reach to the ovule part. 1 or 2 they may reach through pollen tube. So here through pollen tube 2 male nuclei they comes and attaches to the ovule at the basal part, which we are calling it as micropylar region. And the female gametophyte, you see that it is having 7 cells with 8 nuclei. And basal part, if you observe it, egg cell will be in the center. Either side of that we are able to find the helper side cells or the synergids. They help that first male gamete to fuse only with the egg cell. That is what the help they are rendering. That is what they were called as helper cells or synergids. In the center of the embryo sac, we are able to find polar nucleus or the secondary nucleus, which is diploid in nature. 2 sets of chromosomes will be there. At the top, we are able to find the antipodals. 3 cells are there. They are called as antipodals. Next here, in this diagram, we are able to find 1 pollen grain with the help of pollen tube. After germination, 2 male gametes they reach to the ovule. And here the first male gamete fuses with the egg cell. Then each one, egg cell and the male gamete, both male nucleus, both are having the haploid nuclei. So the diploid zygote will be formed. So this comes under first fertilization. Second male gamete fuses with the polar nucleus, and then Polar nucleus already it is having two sets of chromosomes, and the male nucleus it is having one set of chromosome. Haploid plus diploid comes under triploid. So endosperm nucleus is triploid in nature. And at the top two antipodals will be there. Now let us know about the seed germination. Seed germination. When the seed is having the seed coat. Just before diagram once again let us see. Here we are able to find the different parts in this ovule structure. This is what the stalk of that is called funicle, and outer integument and inner integument they will be the future seed coats. They will be the future seed coats. The cells which are there inside the ovule, that is what we are calling it as nucellus. And from where the integument they start coming, that antipodal side we are able to find chalaza. Okay. Next comes to seed germination. When the seed coats, some of the seeds they are having single seed coat, some they are having two seed coats. When favorable conditions are there, when the soil is having some sufficient quantity of water, the seed coat absorbs the water from surrounding area and becomes smoothened and it bursts open. And we are able to find the embryo development. Later on, the seed coat bursting and the baby root comes out. Here, cotyledons, in between the cotyledons, we are able to find a small rod-like structure which is having epicotyle and hypocotyle, two parts will be there. Next, in the fifth diagram, you see that they are having foliage leaves. They started developing until the leaves they make their own food. These two cotyledons they supply the food and nourishment to the growing plant. So that is what we have observed, that the sixth diagram it is having two widely opened foliage leaves. Next, let us see that life cycle of a flowering plant. If it is the datura flower plant or any other plant, you see that when the mature plant is there, adult plant is there, it start bearing the flowers. And the flower is having bisexual flowers like datura, it is having androecium and gynoecium. Next, whenever the pollen grains they reach to the stigma of the flower, then the pollen tube will be formed, and then pollen tube, through pollen tube, male nuclei they enters into this particular embryo sac and double fertilization occurs. After fertilization, zygote will be formed. First fertilization after zygote will be formed. That zygote is diploid in nature. Later on, it develops into embryo, and you know that whatever the swollen part of the ovary, it changes into fruit, and the ovules only changed into seeds. So after that, when the seed germinates, you are able to find the baby plant seedling. Next, after that again it grows. The cells which are there in that seedling divides and divides, and a mature plant will be developed. The cycle repeats. So this is what the life cycle of a flowering plant like datura. Now let us see here cell division and continuation of life. Here Virchow, Rudolf Virchow, who we have learnt about this name in the 9th standard itself. Cell theory was proposed by actually Scladen and Swan. One is botanist and the other one is zoologist. And here he has proposed one phrase called omnicellula and decellula. Next, cells they arise from, normally the new cells they arise from pre-existing cells. Later on another scientist Robert remarked, he told about in the smaller organisms or unicellular organisms like paramecium and all, binary fission, how it will be taken place. One parent cell divides into two daughter cells. In the same manner, in the matured cells also, in the other organisms also, there is a mitotic, there is a division. Cell division will be taken place. Cells they will be multiplied. Next, Walter Fleming. Mitos means here fine thread-like structures, and these strings they divide longitudinally. He observed the strings in the cells, in the nucleus of the cells, they started dividing. So they were later named as chromosomes. Chromo means colorful bodies. Next, after that, Wilhelm rocks. Wilhelm rocks. Chromosomes only, they carry the genetic material or heritable material from one generation to the other generation. Parental to the parent cell to the daughter cells. Later on, August Weismann hypothesis, in that successive generations, individuals they are having same number of chromosomes. Next, later on, Theodore Boveri scheme of mitotic division he proposed. So after that, if you take this, if you observe the cell division, we are able to find here the sperm cell which is obtained from the male organism and the egg cell from the female organism. These two, they fuse together to form the fertilized egg. In our human body, if you observe that, if you take the blood or the glands like exocrine gland like liver and pancreas, and mixed gland like pancreas and all nerve cells, and the skin, muscles and bones, they are all having diploid set of chromosomes. So here we are able to find that after repeated divisions in the zygote, these different cells which are combined to form the tissues combine to form the organs combine to form the organism. So we are able to find again that organism, if it is male organism, they are ready to release the spermatozoa after meiotic division. After meiosis, we are able to find the chromosomes number actually 46 in human beings race, and those 46 chromosomes they enter into the sperm cell after reduction division only 23. When in the female also the same kind of reduction division takes place, and in the egg cell only 23 chromosomes only will be there, and 23 plus 23 male gamete called sperm, when it fuses with the egg cell with 23 chromosomes, again zygote is having 46 chromosomes. In this way we are able to find the 46 chromosomes in all the cells in our body. Next, if you take the cell division, it is having a resting phase called interphase. In the mitosis or the equal division, before it starts, there is an interphase which is having different stages. Gap 1 phase, here it is lasting for nearly 10.5 hours. Next one, synthesis. Synthesis means the DNA synthesis will be taken place, that also 10.5 hours it will take. And G2 phase means gap 2 phase, it is in between synthesis and mitotic division, so that lasts for 3.5 hours. And mitosis, it takes nearly 1 hour. So this is what we are calling it as cell cycle. Later on, let us discuss about the mitosis. In that, karyokinesis and cytokinesis, two phases will be there. Karyokinesis means only the cell division which occurs only inside the nucleus. So here, inside the nucleus, how the changes will occur when the mitosis is taken place during 4 phases called prophase, metaphase, anaphase and telophase. In the prophase, you see that the animal cell, they consist of centrosome with 2 centrioles, they are going to the respective poles, and the chromosome they become shortened, condensed form, and clearly we can able to visualize under the microscope, prophase stage. And in the metaphase stage, you are able to find when the centrioles are reaching to the 2 poles, from there onwards some spindle fibers they start coming, and they are attaching to the center of the chromosome, which we are calling it as centromere, and they are connecting to the other pole where the other centriole is present. In the third phase, we are able to find the anaphase, the spindle fibers they contract, and these chromosomes they divide half, and chromatids they are reaching to the respective poles, they are resembling like letter V or J. And the next phase is telophase. Around the chromatids again the chromosomal material will be added and they become chromosomes. Around the chromosomes, again nuclear membrane will be formed. And there is a small constriction for the membrane, and then it deepens, which divides the cell into two equal parts. After this karyokinesis, there is a cytokinesis. Cytokinesis means cytoplasm division. So, after karyokinesis, cytokinesis will be followed, and each parent cell divides into two daughter cells with the same number of chromosomes, and whatever the organelles present in the parent cell, equally they were distributed to the two cells. This is what the mitosis which occurs in our somatic cells in our body, except the testes in males, except the ovaries in female. The rest of all the cells, for repairing time or growth and development time, this mitosis will be taken place. It plays a very key role. Thank you, children.